Hello everyone, today I'm going to introduce you a biological process called signal transduction of insulin. It's a biochemical pathway by which insulin increases the uptake of glucose into fat and muscle cells. First, we have a phospholipid bilayer membrane that divides the cell from the rest of the world. In between the phospholipid, embedded an insulin receptor, which is basically a protein. When detecting the increase in blood glucose level, insulin is produced by the pancreas in a region called islets of Langerhans. The insulin then binds to the insulin receptor embedded in the cell membrane of a muscle, fat, or brain cell due to their complementary structures. Before we go on, let me introduce a molecule called ATP, which is composed of 1 adenine, 1 ribose, and 3 phosphate groups. One phosphate group will be clipped from the ATP and added to the intracellular domain of the receptor, thus activating the receptor. We call this process autophosphorylation. The activated receptor protein acts as a kinase, which is an enzyme that catalyzes the transfer of a phosphate group from ATP to a specific molecule. Then, the insulin receptor will add a phosphate group from ATP to another closely associated kinase, and another. Maybe one more. And then the phosphorylation event will go on and on and on to pass down the signal corresponding to the level of blood glucose from outside to inside of the cell. And all these kinases are unique in nature with their own name and structure. However, I'm not going to overwhelm you with all the skeletons, and there are over 60 of them. And we call this kind of flow of event cascades. And now I need to introduce the GLUT4 protein, which is basically a glucose transporter embedded in the GLUT4 storage vesicles, which allows the glucose to travel into the cell without interacting with the hydrophobic cell membrane. And the blue incomplete circle here represents the RAP protein. What is special about it is that it interacts with the motor protein necessary to move the vesicle, causing the motor protein to be in an inactive state. This hence keeps the GLUT4 storage vesicles in a cyclic state near the cell membrane, unable to move around. The final step of this cascade involves the phosphorylation of a protein that activates the RAP protein, preventing it from interacting with the vesicles. GLUT4 storage vesicles are then translocated and embedded GLUT4 on the cell membrane. This allows large quantities of glucose to enter the cell, either being used during respiration or stored as glycogen. Through the insulin signal transduction, the body will lower the blood glucose level, keeping us healthy from diabetes by homeostasis. Thank you for watching this video.